Hello. Have you ever turned on Zoom and you realize that you haven't looked at yourself yet today? You turn on Zoom and you're like, ah, that's what happened just now to me. So maybe I'll hide away in my little hood because the Zoom touch up's not even helping. All right. So we are going to finish up chapter 12, mitosis, cell cycle. Mitosis, cell cycle. Mitosis, cell cycle. Okay. Let me share. Okay, let's do this. All right, so chapter 12, we talked about mitosis, why we need mitosis. Oh my God, I'm sorry, I've got to just scroll through this. We talked about chromosomes. Remember the stages of mitosis. We have interphase, which is G1, S, and G2. GS is really important because that's when you get DNA replication. We also get replication of our centrosomes. Interphase is 90% of a cell's life. Most of a cell spends their time in interphase. Prophase is next. That's where you start to see chromosomes condense into these individual chromosomes that you can see. Prometaphase, we start to see the mitotic spindle, which are again microtubules growing out of our centrosomes on either side. Metaphase, we get chromosomes lining up at this metaphase plate with our centrosomes forming the mitotic spindle between. And remember, there's a real push and pull of these microtubules trying to line them up. Anaphase, they're going to pull, sister chromatids are going to pull apart. Telophase, we start to see nuclear envelopes reforming. Cytokinesis, when we get actual cytoplasm splitting, whereas mitosis means nuclear division. All right. And you should be able to identify these by microscope pictures or by diagrams. And in fact, you should be able to draw them as well. And I've posted a screencast all about drawing mitosis as well. Okay, so we're starting now with microtubule spindles. They are one of the most beautiful things to watch. I will show you some movies in class, but you're gonna have to remind me to do that. But they're dynamic. They're moving, and if you stain them with certain things, so for example, in this diagram below, we have a DNA stain in blue, microtubules in green, and the kinetochores in red. Remember that when we have a chromosome, so here is an individual chromosome, remember that in S phase, they get duplicated to create sister chromatids that are held together at the centromere. Remember that the kinetochores are protein complexes that get built on the centrosome, on the centromere, so that microtubules can go and attach to them. All right. So they have these long microtubules that form from the centrosome that reach out and try to find the kinetochores. So you should be able to recognize all of the things on this picture. You should be able to recognize the centriole pair, a centrosome, a duplicated chromosome composed of two sister chromatids, right? There are two kinds of microtubules. We have aster microtubules, which kind of form a star all the way around the centrosome. So here, let me do it in white here. So that would be all of these microtubules here that I'm pointing out with the white. Those are the aster microtubules on both sides. Aster microtubules, and called that because they look like a star. And then we also have microtubules which will go and bind to the kinetochore, all right? And those are called kinetochore microtubules, all right? I'm not gonna make you know about the other one. Okay, so this is a really cool experiment. Anyway, I'll tell you what's going on here. This is beyond the scope of normal AP Bio, but I think it's freaking rad. And I like to show you actual data, all right? So here is the experiment. They used a cell undergoing anaphase. Remember, that's when the sister chromatids are pulling apart. And what they do is they mark the microtubules. So you can see that here. I'll label the pictures 1, 2, and 3. Number 2 here, they're marking the microtubules, and they see what happens to that mark. And if you see in number 3, what's happening is that the chromosomes here are actually getting closer to the mark, but the mark is staying equal distance from the centrosome, right? So what does that mean, right? And what it means is that the microtubules, instead of like disassembling from the chromosome, they're actually disassembling at the kinetochore, right? So the question, the experiment was aimed to ask, which end do these spindle microtubules shrink from or depolymerize from, right? The poles or from the centrosome, the poles or from where they bind the chromosome. And it turns out microtubules shrink from where they bind chromosomes, and that is at the kinetochore, which is again a group of proteins that assembles at the centromere, where those sisters were connected, right? All right, so now we're talking about the cell cycle. So different cell types have different cell cycle control, and that makes sense. So think of your skin cells. They have to divide more rapidly because you're constantly sloughing off skin cells. If you're a liver cell, you divide only when necessary, only when there's damage or something like that. And then we have certain cell types like neurons, which once they reach maturity, they don't ever divide again. And that's why these neurological diseases like Parkinson's and Alzheimer's are so detrimental, because we can't figure out a way for those neurons to divide again once they reach maturity, for the most part, right? So the cell cycle is controlled by several different things. We have external cues and we have internal cues, right? So for the external cues, they are anchorage dependence, meaning they need something to kind of sit on or grow on, right? So when they're anchored down to something, that kind of helps. Also density dependent. So our cells in general don't like to grow on top of each other, right? So for example, we usually have a nice thin layer of cells rather than a big clump of cells. And then we have internal cues, where we have control systems, molecular control systems, which we'll talk about, cell cycle checkpoints, and growth factors. All right, so let's look at this figure. So cells, if you grow them in culture, meaning you take cells from, for example, a human sample, and you draw them and you grow them in a dish in the lab. And we would do this at 37 degrees Celsius, which is the temperature that our human bodies are, all right? And I've done this lots of times in lab. Human cells are very easy to grow. And we can talk more about this, but many times they're called HeLa cells, and they were collected from a woman named Henrietta Lacks. And they're like the most successful lab human specimen ever in history, but the way they were collected was very unethical. It's a very interesting story. If you haven't read The Immortal Cells of Henrietta Lacks, it's cool reading. All right, so cells like to anchor to the dish, and then they divide, right? So they like to anchor down. That's the anchor dependence. When cells have formed a complete single monolayer, right? They stop dividing, right? And then that's the density dependence. So those are the two kind of external cues, right? So if you scrape some cells out, the cells will kind of go through like a wound repair, and they'll fill in that gap. Okay, but then they'll stop. However, we know that cancer cells do different things than normal cells, and cancer cells basically break all of the rules. So they don't need a big, like a surface to grow on, which kind of can result in metastasis, when cancer cells kind of go away and go to other body parts. And they can also form on top of each other, and that can create a tumor. So if these external cues are not followed, then you can result in cancer. Right? So cancer cells basically break all of the rules, all right? Normal cells, if there is some sort of DNA damage, then normal cells stop the cell cycle. They try to repair the DNA. If they can't, they will actually kill themselves, right? And that's a process called apoptosis. And I always remember that because I think of the cells popping or opening up, right? Cancer cells, if they have unfixable DNA damage, the cells don't stop dividing. They just continue to divide and divide and divide, right? And that can cause cancer. And all cancer is, by definition, cancer is uncontrolled cell division. That's it. So if you have uncontrolled cell division, then you can have a cancer. You can have cancer. And there's lots and lots and lots of different types of cancer because there's lots and lots of different types of cells in our bodies that can have this uncontrolled cell division, right? All right, so this brings us to more the internal cues, all right? So first we have cell cycle control, all right? And there's two main things, right? We're gonna be talking about cell cycle checkpoints over here, the first one, and then we're gonna talk about regulatory proteins, number 2. Right, and I'm going to go over this pretty fast, but there's a lot of detail, which I'm not going to get too crazy about. All right, so those are the two that we're going to go over. The first one are these cell cycle checkpoints. All right, so there are 3 checkpoints in the cell cycle. Right, and those checkpoints are meant to prepare and make sure a cell is ready for division. Let me move my face. All right, so number 1. It's called the G1 checkpoint. And it asks, is the genome and the environment okay for cell or for DNA replication, right? So I always think literally there's a little dude with a stop sign. It's actually a little protein instead of a little dude, but that stops, says, hold on, wait for a second. Is a genome okay? Is the environment okay? If everybody looks good, I will let you go through. Right? So as a cell goes along, it can stop there and it says, hey, are you ready for DNA replication? If so, we're going to go through DNA replication. And then we have a second cell cycle checkpoint called the G2 checkpoint. And this is right before mitosis. So it's going to ask, hey, was your DNA replicated correctly? Are you ready for mitosis? Right? And so if your cell is like, yep, DNA replication went okay, we are ready to start nuclear division, then your cell will continue. And we have a third cell cycle checkpoint called the M checkpoint or the metaphase checkpoint. And this makes sure that at metaphase, are your chromosomes or your sister chromatids ready for separation? At metaphase, all lined up, ready to split, question mark. And if they're not, then we have a little dude here, or in the cell's case, a little protein that's like, okay, then we're not gonna split. We'll hang out in metaphase for as long as it takes. Otherwise, we're gonna stop and abort mission, all right? So if the cell is stopped at a checkpoint, it can do 3 different things. It can stop, wait, try to repair the situation. So for example, during metaphase, if one chromosome pair is way out here, the middle metaphase plate is here, you can either wait for that sister chromatid to get in line with the others, or you can exit the cell cycle, or you can induce cell suicide or apoptosis, all right? So there's these 3 cell cycle checkpoints, one at G1 to S transition, make sure you're ready for DNA replication, one at the G2 to M transition, make sure you're ready for mitosis, and then one at the metaphase to anaphase transition, make sure you are ready to split your chromatids. There's also cell cycle regulatory proteins, all right? And I'd like to show you actual experiments. So here is an experiment. They were using Xenopus, which is this crazy looking frog that gets, so this is a female frog, and she has given a lot of eggs. And these eggs are not coming out of her mouth, even though they look like it. They came out of the frog's cloaca. I think that's what it's called. Cloaca? Yeah, cloaca, I think, in frogs. And it has a lot of eggs there. All right. So these were conducted on these single eggs. So what they did is that researchers injected the cytoplasm from cells that are in mitosis into an egg. And what it did is it caused the cell to form a spindle. So what it did is it drove the oocyte or the egg into metaphase, right? So there's something in mitotic cytoplasm that can cause cells to start mitosis, right? If you do a control experiment where you inject the cytoplasm from an interphase cell, remember interphase would be G1S or G2, not mitosis, into an oocyte, nothing happens. So here, no mitosis, right? So there is something in here, I'll put this here. There are proteins present. They later determined that they are proteins. There's some proteins present in mitotic cells that can induce mitosis in other cells, right? They called that, and they didn't know what it was called. They didn't know what it was at the time. They called that maturation promoting factor or mitosis promoting factor, right? We now know what that promoting factor is. We're not going to go into too many details about it, but there are two really important proteins here. We have a protein that cycles in the cell cycle. So here we have the cell cycle, and that's shown here with the green, blue, right? We have interphase, mitosis, interphase, mitosis, interphase, mitosis. And cyclin concentrations cycle in the cell cycle. They go up and down, up and down, up and down. And when their levels get very, very high, they activate another protein called CDK, right? And that's called a cyclin-dependent kinase, right? And we haven't really talked about kinases yet, so we're not going to go into too much detail. But cyclin levels, as they go up, they induce another protein, and that protein can thereby induce mitosis, right? So here, let me put this. I don't like the way I did that. So cyclin levels up high. When cyclin levels high, that induces CDK activity, and that drives mitosis, right? All right, we're not going to talk about all the details of that. But here is just showing this again, that as you increase these cyclin levels, then you are increasing your CDK activity, and that drives mitosis, all right? And so if you look at the cell cycle in this figure over here, you can see, again, as cyclin levels accumulate, what that does is that it activates our CDK. And again, we're calling that maturation promoting factor. And that literally is just the stuff in mitotic cytoplasm that drives mitosis, all right? And then cyclin gets degraded, and that's why we see this crash, right? There's a lot of crazy details in here, which if people want to know about, I'm happy to share, but I'm not going to do that now. All right, and then we're not going to talk too much about this, but I just want to again point out that cancer can really, because cancer screws up the cell cycle, right? And we will talk about these another time, but there are different types of cancer cancer cells, but sorry, there's different ways that you can cause cancer in a cell. But again, cancer, all it is uncontrolled cell division, right? So very often what you're doing is you're screwing up either these cell regulatory proteins or these cell cycle checkpoint proteins, right? And if you can do that, if you do that, then that will cause your cancer, all right? And I'm gonna hold off on talking about P53 and RAS. They're awesome. We will hopefully talk about them when we get to cell signaling, but we just don't have time in right now, all right? Okay, I will stop there. Again, there's lots of questions here that you should take a look at. All right. These kinds of questions are going to be essential that you understand what these drawings mean, what part of the cell cycle you're in, etc. All right. All right. Chip, chip, cheerio. Toodles, poodles.